Okay, so we record.
Am I audible, Pa? Veronica? Will I get one yes? Yes, am I audible to you all? Shall I start the class for you all? Will I get one yes from any of you? Tarunika, Vivek? Am I audible? Yes, very good evening, Shubhan. And yes, Kavya, am I audible to you? Can I get one yes? Am I audible? Shubhan? Very good evening, Shubhan. How are you after a long time? Yes, thank you, Shubhan. Thank you for that message. Yes, Kavya, very thank you. Yes. So let us discuss respiration in plants. Okay. So this chapter, as you all know, is a very simple chapter, and but ATP related questions we may get from this chapter. Okay. So we have to be very cautiously learning about respiration and ATP, number of carbon and phosphorus. Okay. So before we start, can you tell me, children, the respiration, is it anabolic or catabolic process? Is it anabolic or catabolic respiration process? Anybody, can you answer me? Respiration, is it anabolic or catabolic process? So as we all know, respiration is catabolic process and it happens in cell. So it is even called cellular respiration. Okay. So in a cell, of course, it takes up the glucose. So assume this is a cell. It takes the glucose. Okay. So when it takes the glucose, then it process and release two process things. One is carbon dioxide, another one is energy. So this is what we call it as respiration. As it happens in cell, it is called cellular respiration. and we break down the process. I hope you are clear. Yes? Very good. Now, photosynthesis is anabolic process and respiration is catabolic process. Okay, fine. Tell me. So, breathing is a demanding process for animal. Is it a very demanding process for plant? Yes or no, children? Yes, Kavya and Subhan. Yes. So it is actually not that demanding process in plants. Okay. So you know what are the organs which are helpful in breathing in plants? What are the organs that are helpful in breathing in plants? Is it not stomata? Is it not lenticles? As you all know, stomata we found in leaf and lenticles we found in stem. Okay, so these openings help the plant to breathe. It is not highly demanding procedure. Yes. Very good evening, Jeevan and Shalini. Very good evening. We just started, Shalini and Jeevan. Yes, respiration in plants. I'm just giving introduction. So the organs in plants are stomata and lenticles are helpful for breathing. Okay. Now for respiration process, there are four major process involved. Can anybody tell me any of the process involved in respiration? What are the steps involved? Of course, it is glycolysis. And second one is oxidative decarboxylation. Third one is Krebs cycle. Fourth is electron transport. Can anybody answer me among the four processes where we produce more ATP? So in which process we produce more ATP comparatively? Hi Tilak. Very good evening. Yes, my dear Cody sponsor. So tell me, Tilak, where we are producing more ATP, is it in glycolysis or oxidative decarboxylation or Krebs cycle or electron transport? Where we produce more ATP? Yes. Let me see who is answering first. More ATP we produce. Possible question. From 5, 4, 3, 2. Yes, Abhishek. Very good evening, Abhishek. Thanks for participating, Abhishek. Just we started respiration in plants. It will be useful. Important chapter. ATP related questions will come. Yes, excellent answer by Jeevan. Yes, Jeevan, it is. It is ETS, electron transport. More ATP is produced during electron transport system. Hi, Anston. Very good evening. Thanks for participating. Very happy to see your name. Thank you so much for your participation. So, more ATP we are producing in electron transport system. Yes. So, let us see how the process is going to happen one by one. So, shall we start with glycolysis? Yes. So, glycolysis, is it a complete oxidation process, incomplete oxidation or partial oxidation? Yes, Subhan, it is electron transport. Very correct answer. Excellent. Yes, Malay Vanakkam Jeevan. Malay Vanakkam. Tell me, is it a complete oxidation or incomplete oxidation or partial oxidation? So, glycolysis, first possible question. Is it complete or incomplete or partial oxidation? Yes, anyone? Shall I reveal? Yes, it is partial oxidation. Yes, excellent answer by Jeevan again. Yes, it is partial oxidation, Jeevan. Partial oxidation. Because there is no evolving of carbon dioxide in any of the 10 steps. So, it is partial oxidation, no evolving of carbon dioxide. Okay. Now, is it aerobic or anaerobic or both actually? The process is aerobic or anaerobic or both? Actually, it is both. So, it is aerobic and anaerobic process. Okay. Now, where it will be carried out? Will it be carried out in mitochondria or it is carried out in chloroplast? Yes, Tarunika, excellent. It is both. Where it is carried out? Is it carried out in chloroplast? Yes, Jeevan is excellent. It is both. Is it in mitochondria or it is in chloroplast or it is in cytoplasm or nucleus where we process glycolysis. Hi Yogeshwari, very good evening. Yes, both is very correct answer. Yes, cytoplasm, excellent Jeevan again. Yes, it is happening in cytoplasm. Okay, so cytoplasm is the correct answer. So where exactly the glycolysis is carried out, and you know there are 10 steps involved in the process. Yes, it is cytoplasm, very correct answer. So 10 steps are going on in this process. Okay, and glycolysis is otherwise called EMP pathway. Why it is called EMP pathway? Because it is based on Emden, Mayrov and Pranas. These are the scientists who involved in the EMP pathway and studied this EMP pathway. So it is called EMP pathway or glycolysis. Happens both aerobic and anaerobic condition. Of course, it takes place in cytoplasm children. Fine. So let us learn the 10 steps very slowly. I will proceed. Any doubt, please feel free to ask me. Fine. Now, first step of glycolysis, where we convert glucose into glucose 6-phosphate. Okay, so you know glucose is a 6-carbon component, and when glucose is converted into glucose 6-phosphate, we are adding phosphate to it. How do we add phosphate? From where we get phosphate? Of course, it is ATP. So, ATP will be broken into ADP plus phosphate. So, that phosphate is added on the 6th position of glucose carbon. Wherever ATP is coming, we will get the enzyme called kinase. So, the enzyme is exokinase. This is the enzyme. I hope you are all clear with the first step. Any doubt? Yes. So glucose is converted into glucose 6-phosphate. Now, next step, if you see, this glucose 6-phosphate is converted into fructose 6-phosphate. Thank you, Vivek, for your answer. Yes, fructose 6-phosphate. Okay, so that glucose and fructose are having how many carbon? Is it 5, 4 or 6? Both are identical 6 carbon. So what is the enzyme that involved in the interconversion? They are optical isomers. So the enzyme will be isomerase. Isomerase is the enzyme which convert glucose 6-phosphate to fructose 6-phosphate. I hope you are clear, children. Glucose is also 6 carbon component. Fructose is also 6 carbon component. And both are having, yes, very good, excellent answer. Yes, both are having 6 carbon. Both are having 6th position phosphate. And so the enzyme is isomerase. Now next one, this fructose 6-phosphate, we are going to convert into fructose 1,6-diphosphate. So we are going to convert fructose 1,6-diphosphate. Means we are again adding phosphate in the first position also. Okay, I already told you from where we are getting the phosphate from ATP. So, you know ATP will be converted into ADP plus phosphate. That phosphate is added in the first position. Wherever phosphate is coming, of course, kinase enzyme should come. So, the enzyme is phosphofructokinase. I hope you are all clear up to this. Can anybody tell me up to this how many ATP has been utilized, children? Is it 1, 2? Yes, excellent, Subhan. Excellent. Yes, ATP we are utilizing. And how many ATP we have utilized so far among these steps? Is it 1, 2, 3 or 4? How many ATP has been utilized now? Yes, Tilak. Excellent. My sponsor of Cody. Yes, Jeevan, yes, it is 2. Yes, Pa, it is 2. Yes, it is 2. We are, yes, Subhan, excellent. It is 2 ATP we have utilized so far. So, we didn't generate any ATP, but we have utilized. Yes, Yogeshwari, it is 2. So, I hope you are all clear. Now, that fructose 1,6-biphosphate, we will be converting into 2 components. It is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So these three are having how many carbon, any of you? This dihydroxyacetone phosphate and glyceraldehyde 3-phosphate has how many carbon atom? The enzyme we use is aldolase. Of course, how many carbon atom is the question I asked. Now, here also they are interconvertible like glucose and fructose as we discussed. They are mirror image. Yes, 3 carbon, excellent answer by Tilak as well as Jeevan, excellent answer. Yes, it is 3 carbon component. Yes, Yogeshwari, it is 3 carbon component and the enzyme here is isomerase again. Yes, I hope you are all clear. Anyway, so you can just see from here also. So we are breaking the glucose catabolic process is happening as we discussed and we release carbon dioxide and ATP cellular respiration. As we discussed, photosynthesis is anabolic. Respiration is catabolic process. And breathing happens in plant either through stomata or through lenticles. So EMP pathway we are discussing. As we discussed glucose to glucose 6-phosphate, we are adding 1 phosphate on the 6th position. Interconversion of glucose 6-phosphate to fructose isomerase enzyme. Fructose 1,6 bisphosphate. Again ATP is used. And we form 2 component, that is 3 carbon. Up to this we have finished. Yes. Now we have to continue from here. Now, the next one, what happens? Let us continue from one side of the component, either glyceraldehyde 3-phosphate or dihydroxyacetone phosphate. So, this will be forming what? This will form 1,3 Hi Shrikant. So happy to see your name. I am so fine after a long time. Yes, Kathleen. Very good evening, Kathleen. How are you? Happy to see your name, Kathleen, my dear BTS. And Shrikant, very happy to see your name. So 1,3 bisphospho glyceric acid. So 1,3 bisphosphoglyceric acid, how we form? Already we have 1 phosphate. Dihydroxyacetone phosphate. 1 phosphate is there. 1 more phosphate we have to add. But we are not adding through substrate phosphorylation. That is not by ATP. Then how do we add? See, it is not mentioned in our NCRT. Yes, very good evening, Parthasarthi. We just started glycolysis, Parthasarthi. So, like you can see, it is inorganic phosphate PI, which is not mentioned even in NCRT. But we may get confusion how this phosphate is doubled. 1,3 bisphosphoglyceric acid. And this step, even reduction process is happening. So we are converting NAD into reducing power is generated. NADH plus plus. Either we can write NADH plus plus or NAD2H also. So please remember, this is possible block actually. Very good, excellent. So yeah, next point I wanted to say that it is reduction and phosphorylation. Yes, Jeevan. It is reduction and phosphorylation. Question will come. Which place we are having reduction and phosphorylation both happening? It is the conversion of glyceraldehyde 3-phosphate into 1,3 bisphosphoglyceric acid. So as you know, reducing power is generated, it is reduction. Phosphorylation because 1,3, one more phosphate is added. So NAD is reduced to NADH++. Can anybody tell me, 1 NADH++ is equal to how many ATP? 1 NADH++ is equal to how many ATP? Is it 3 or 2 or 4? Yes, children. Yes, it is 3. Jeevan, excellent answer by you again. Yes, it is 3. So we are forming 3 ATP from 1 NADH++. Yes, Yogeshwari, excellent answer by you. It is 3. So now 1,3 bisphosphoglyceric acid, we will be converting into 3 phosphoglyceric acid. So one phosphate is missing now. Okay. So we have to tally that phosphate. So how that phosphate is utilized? We will be synthesizing ATP. So from ADP, that phosphate will combine and forms ATP. And I told you wherever ATP is coming, it is kinase enzyme. So the enzyme is phosphoglycerate kinase. So this is the enzyme which is involved there. Phosphoglycerate kinase. Kinase, wherever ATP is there, phosphoglycerate, you can take it from the prior product. And here the enzyme is, as the reduction is happening, phosphoglycerate dehydrogenase. I hope you are all clear up to this. Yes, 3 is very correct. Excellent, Tarunika. By you also. Yes. Now, this 3-phosphoglyceric acid will be converted into 2-phosphoglyceric acid. Means what? We are just changing the position. From third place, the phosphate is going to the second place. So, it is 2-phosphoglyceric acid. So what may be the enzyme? Here we are not reducing any phosphorus. Phosphate is only one, phthalate, 3-carbon phthalate. So the enzyme is phosphoglycerate mutase. It is only an exchange of the phosphate. Phosphoglycerate mutase is the enzyme. Okay. Now this 2-phosphoglyceric acid will be converted into phosphoenol pyruvate. Phosphate is there, 3-carbon component is there. Okay, then what is happening here? Here important step, the enzyme is enolase. Enolase means what? We will be removing water. Can anybody answer me? Enolase, wherever it comes, do we remove carbon or oxygen or water? What do we remove when we have enolase enzyme? Yes, any of you? When we use enolase enzyme, yes, excellent answer by Jeevan. Yes, it is water. Okay, we will be removing. Water is removed. H2O. Okay. Now the last step, phosphoenol pyruvate, we will be converting into pyruvic acid. Again a 3-carbon component only. But what is missing? Phosphate. Pyruvic acid we form. Okay. So we form pyruvic acid, which is a 3-carbon component, but phosphate is missing. So what will happen to the phosphate? We will generate ADP utilized with the phosphate to form ATP. So ATP wherever forms, we use kinase enzyme. So here the enzyme is pyruvate kinase. This is the step. So we have 10 steps involved in this process. And how many steps we produced ATP? How many steps we produce ATP? Is it 1, 2, 3 or 4? How many steps we produce ATP? Yes, Jeevan, excellent answer by you. It is 2. Yes. So 2 steps, 100% correct, Jeevan. It is 2 steps we are producing, as you can see here. 1 ATP we produce here, another one here. Okay, so 2 ATP we are producing. That means if you process this glyceraldehyde 3-phosphate, the same steps will repeat. Okay, so you are producing ATP in 4 points. Yeah, only ATP it is 2, yes. Okay, so 4 places we will be producing and we spend 2 ATP here. So 4 minus 2 will become 2 ATP as such. And NADH in one place. So 1 NADH is equal to 3 ATP. 2 NADH is equal to 6 ATP. So totally we will produce 8 ATP net gain. That's what the total net gain. So net gain of ATP is equal to 8. Okay. So how we tally? 4 ATP minus 2 ATP what we spend will become 2 ATP, and we are producing 2 NADH plus plus, which is equal to 6 ATP. So total is 8 ATP. I hope you are all clear, children. So are we clear with the 8 ATP? So possible questions, of course, this important step is here. So where we have reduction and phosphorylation, where we synthesize NADH++, possible question. Where water is removed, another possible question. Okay. So you can see even here. So what we discuss, NADH++, inorganic phosphate will be added in this step so that we are getting 2 molecule of bisphosphoglyceric acid will be converted into 2-phosphoglyceric acid and we form phosphoenol pyruvate. Then it forms pyruvic acid on the 10th step. So water is removed and ATP form. Overall process. So when does NADH is produced in cellular respiration? A possible question. Can anybody answer me? When we convert phosphoglyceraldehyde to DHAP or phosphoglyceraldehyde to bisphosphoglyceric acid, conversion of bisphosphoglyceric acid to PGA. So you understood the expansion, phosphoglyceraldehyde to bisphosphoglyceric acid. One possible question. Let me see who is answering first. Where we produce NADH++. In which of the step? Yes, 3 is the correct answer. Many of you answered yes. Jeevan, Yogeshwari, Tarunika, yes, Subhan, it is 3. No doubt, excellent answer by all of you. Yes, it is 3. Okay, where we convert phosphoglyceraldehyde or dihydroxyacetone phosphate into 1,3 bisphosphoglyceric acid. Excellent answer. Okay. Now this question, how many ATP molecule are directly synthesized in glycolytic pathway from one glucose molecule? That is from one glucose, how many ATP? One glucose, please remember. It is one glucose they are asking. How many ATP we generate directly? Directly also is a keyword. From one glucose is also a keyword. Yes, I'm waiting for your answer. Yes, Jeevan, excellent answer by you. It is 4 ATP from one glucose directly. Okay, so directly is a keyword. One glucose is a keyword. From one glucose, we generate 4 ATP. Is the very correct answer. Excellent answer by Jeevan. Yes. Yes. Okay. Now this question. How many carbon atoms are there in a molecule of glyceraldehyde phosphate? See, they are asking carbon atom number. Is it 3 carbon, 4 carbon, or 5 carbon, or 6 carbon? Glyceraldehyde phosphate is a how many carbon atom molecule? Yes, Jeevan again answered. Yes, it is 3 others. Yes, Subhan, excellent answer by you. It is 3 glyceraldehyde 3 phosphate is a 3 carbon component. So fructose will break into glyceraldehyde 3 phosphate dihydroxyacetone. Both are 3 carbon component. Excellent 100% correct answer. Yes. Okay. Now we have to discuss anaerobic respiration. I hope you are all clear with the aerobic respiration. Any doubt anywhere, children? Yes, Yogeshwari, excellent answer. It is 2. Yes. So, anaerobic respiration without oxygen. Okay. So, in anaerobic respiration, where the same steps will happen, we are converting glucose into after so many steps pyruvic acid. So, you know the steps involved. Just now we discussed. So, all these steps, pyruvic acid has been formed. Now, this pyruvic acid by yeast, we form what? Ethanol. Now, tell me, children, when we form ethanol, do we form carbon dioxide or not? When ethanol is formed, carbon dioxide is also formed or not? This is the question. Yes or no question? So, we form ethanol. Yes. Are we forming carbon dioxide or not is the question. Yes, we form. Excellent answer by Jeevan. Yes, CO2 will be formed. Okay. And this process, you know very well. Yes, Subhan, excellent answer. Yes. Yes is the correct answer, of course. It is by yeast. And what are the points we need to remember regarding the yeast? I hope you all know what is the yeast which involved in the ethanol formation. Name the yeast which involved in ethanol formation. So, you know, when yeast is in 7%, it is capable of forming alcohol. Where if it exceeds 13 percentage, it may be poisonous to the possessor itself. So, the yeast itself, it will be poisonous. Processor. I asked what is the name of the yeast. Any of you remember what is the name of the yeast? Is it not Saccharomyces cerevisiae? Brewer's yeast. Saccharomyces cerevisiae. Even in microbes in human welfare, it will come. Okay. So it is otherwise called brewer's yeast. So carbon dioxide is formed. Other possible question is, what is the enzyme it will be having for the production of alcohol? Alcohol dehydrogenase is an enzyme. What mineral we are having? Zinc is a mineral we are having, which is involved in alcohol dehydrogenase, or even carboxylase will be there. Yes, children. So this is regarding the process. Now what happens here? You know, here we net gained 8 ATP so far. 8 ATP we have got. We generated already. Yes, zymase also. So these are all zinc based enzyme only. Carboxylase, alcohol dehydrogenase, yes, zymase in formation of alcohol. So we have 8 ATP. Now what happens here in this conversion of pyruvic acid to ethanol anaerobic process, we convert NADH++ into NAD. That means it is oxidized. Do we get any energy, children, when it is in the form of NAD? Do we get energy in the form of NAD? Can we convert into ATP? Yes or no? NAD, does it give any energy directly? No. Is the correct answer Jeevan? Excellent, Jeevan. Okay. So it is reoxidized actually. So we are not getting any energy through that. So loss of 3 ATP per turn means what happens? 6 ATP we may lose. So when we lost 6 ATP, net gain of the ATP in anaerobic process is how much, children? What is the net gain of ATP in anaerobic process? Who will answer me? Net gain of ATP in anaerobic process is how much? நமக்கிட்ட 8 ATP இருந்தது. நம்ம வந்து NADH++ reoxidize பண்ணிட்டோம். நம்ம கிட்ட 2 NADH process இருந்தது. So when you reoxidize, how much ATP will be the net gain? Yes, Jeevan, again excellent answer by you. And Ajas, yes, it is 2. Very correct. Excellent answer by you again. Yes. So we will get 2 ATP as our net gain. Okay. So this is possible question. Now let me ask you one more question. If 100 glucose molecule undergoes anaerobic process, okay, 100 glucose molecule undergo anaerobic process, what is the total ATP we gain? Is the question. So how many ATP we gain when it undergo anaerobic process 100 glucose molecule? Who will answer me? 100 glucose molecule when it undergo anaerobic respiration ஒரு குளுக்கோஸ்க்கு 2 ஏடிபினா 100 குளுக்கோஸ்க்கு எவ்வளோ ஏடிபி வந்திருக்கும்? Yes, come on children. Anybody? ஒரு குளுக்கோஸ்க்கு 2 ஏடிபினா 100 குளுக்கோஸ்க்கு anaerobically how many ATP is the question? Yes, Subhan and Jeevan, excellent answer. It is 200. Excellent, 100% correct. Yes, it is 200. Very correct answer, Subhan and Jeevan. Okay, so it is 200 ATP we are net gaining. இதே உல்டாவா கூட கேட்கலாம். இப்போ இப்படி ஒரு question வருதுன்னு வச்சுக்கோங்களேன். 400 ATP is the net gain. Anaerobically, na. How many glucose has been used? How many glucose molecules have been used? Yes, again it is 200. இல்லையா 400 ATP என்ன அப்போ எவ்வளோ glucose இருந்திருக்கோ 200 glucose. Excellent answer by Subhan and Jivan. I hope you are clear. Okay, so this is anaerobic respiration, one part of the story. So I'll even show you here. You can see alcoholic fermentation. NCRT picture. Pyruvic acid is converted into ethanol. So you can see the reoxidation of NADH++. So you are having only 2 ATP. Okay. Now, other side of the story, where again we will have the glucose will be converted into pyruvic acid. Same. In the pyruvic acid, anaerobically musclesல என்னவா மாறுது? Musclesல it will be converted into lactic acid. இல்லையா? So it will be converted into lactic acid. Yes. So lactic acid அது மாறிட்டு இருக்கு. இப்போ lactic acid மாறும்போது does it release any carbon dioxide? Does it release any carbon dioxide? Yes, lactic acid. Excellent answer by Subhan. Yes, we are forming lactic acid. Carbon dioxide அதுல வருமா? இது மசில்ஸ்ல நடக்கிறது. கார்பன் டை ஆக்சைடு ரிலீஸ் ஆகுதா? இல்லை என்பது மிக சரியான பதில். எஸ், நோ கார்பன் டை ஆக்சைடு ஹஸ் பீன் ரிலீஸ். ஸோ NADH என்ன ஆகும் அப்படின்னு பாத்தீங்கன்னா NADR ரீஆக்சிடைஸ் ஆயிடும். அகைன் நெட் கெயின் நமக்கு என்னதான் இங்க 2 ATP மட்டும் தான் நெட் கெயின். Yes, Subhan, excellent answer by you. So, idhula in the ATP questions, I hope you are all clear. One more question, let me ask you, see. During fermentation by yeast, alcohol is formed from dash. Is it from protein? Is it from sugar or mucopolysaccharide or fat? Hi, Sanjay. What? Very busy. So during fermentation by yeast, alcohol is formed from proteins, sugars. Yes, Subhan, excellent answer by Sugan. Yes, it is sugar. So we are converting glucose only into alcohol. Excellent answer by Jeevan. Yes. So we finished actually glycolysis process. Okay. இப்போ இந்த கிளைகோலைசிஸ்க்கு அப்புறமா ஒரு சின்ன ஒரு பிரேக், ஒரு குட்டி கதை சொல்லலாமா? Shall I share a small story? Are you ready? Yeah, Sanjay is ready. So the story is about Roger Beniter. யாருக்காவது தெரியுமா யாரு ரோஜர் பெனிட்டர்? லைட்டா. ஸோ ரோஜர் பெனிட்டர் அப்படின்றவர் யாரு அப்படின்னு பாத்தீங்கன்னா, இஸ் an athlete. Okay. So when he was he's an athlete, of course he's a runner, and when he was practicing, அப்போ அவர் ஒலிம்பிக்குக்காக ட்ரைனிங் எடுத்துட்டு இருக்காரு. அப்போ ஒலிம்பிக் ட்ரெயின் பண்ணும்போது யாருமே ஒலிம்பிக்ல 4 மினிட்ஸ்ல கிராஸ் பண்ணதே கிடையாது 1 மைல். ஸோ திஸ் வாஸ் அ ஸ்ட்ராட்டஜி. ஏன் அப்படின்னா நிறைய பேர் கேள்வி கேட்கும் போது இந்த டாக்டர்ஸ்லாம் என்ன சொல்றாங்க, இல்ல இல்ல அதெல்லாம் முடியாது, 4 மினிட்ஸ்க்குள்ள வேகமா ஓடி வந்து அந்த மைல கிராஸ் பண்ணணும் அப்படின்னு பாத்தீங்கன்னா, அப்ப என்ன ஆகும், ஹார்ட் எல்லாம் வேகமா துடிக்கும், லங்ஸால வேலை செய்ய முடியாது, the person may get collapsed. ஸோ அது முடியாது, இட் இஸ் நாட் அட்வைசபிள் டு ரன் வித்தின் 4 மினிட்ஸ் இன் அ மைல் அப்படின்லாம் வந்து they are telling. ஸோ அதனால 4 மினிட்ஸ் இன் அ மைல் அப்படின்றது வந்து பெரிய கொஸ்டின் மார்க்காவே இருந்தது எல்லாருக்குமே. அப்போ வந்து இந்த ரோஜர் பெனிட்டர் சொல்றாரு, ஏன் இதை கிராஸ் பண்ணக்கூடாது, நான் கிராஸ் பண்றேன், வித்தின் 4 மினிட்ஸ்ல நான் வந்து 1 மைல் கிராஸ் பண்ணுவேன் அப்படின்னு சொல்றாரு. இல்ல அப்ப எல்லாரும் சொல்றாங்க, இன்க்ளூடிங் இஸ் ட்ரெயினர்ஸ் டெல்லிங், நோ நோ அதெல்லாம் ரொம்ப விஷ பரீட்சை, அதெல்லாம் செய்யாதீங்க, இந்த மாதிரி பண்ணக்கூடாது அப்படின்னு சொல்றாரு. ஆனா அவர் வந்து கீப்பிங் ஆன் டாக்கிங் டு ஹிம்செல்ஃப், என்னால முடியும், நான் பண்ணி காமிப்பேன், நான் கிராஸ் பண்ணுவேன் அப்படின்னு சொல்லிட்டு அவர் கண்டினியூஸா பிராக்டிஸ் பண்றாரு. அண்ட் யூ ஒன்ட் பிலீவ், he is the first person cross within 3 minutes 59 seconds. Of course it is not crossed 4 இல்லையா? அப்ப அந்த பெரிய பேரியர் உடைச்சவரே அவர் தான், ரோஜர் பேனிட்டர். அதுக்கப்புறமா வந்தவங்க எல்லாருமே வந்து கிராஸ் பண்ண ஆரம்பிச்சாங்க. அதுக்கு முன்னாடி யாருமே ட்ரையே பண்ணல. ஆனா அவர் பண்ணதுக்கு அப்புறம் ஒரு 9 மெம்பர்ஸ் கிராஸ் பண்ணாங்க. அதுக்கப்புறம் 270 ஆச்சு. the number is increasing. So 4 minute என்ற பிரேக் உடைச்சவர் யாரு அப்படின்னு பாத்தீங்கன்னா, ரோஜர் பெனிட்டர். இது எதுக்காக உங்களுக்கு இந்த கதை அப்படின்னு பாத்தீங்கன்னா, சப்கான்சியஸ் மைண்ட். நம்ம மனசுக்குள்ள நம்ம எதை பதிய வைக்கிறோமோ அது நமக்கு கண்டிப்பா நடக்கும். நீங்க ஒரு ஷிப்பை யோசிச்சுக்கோங்களேன். ஷிப்ல நமக்கு மாலுமி இருப்பாரு, அவர் வெளியே இருந்து பாப்பாரு, ஏதாவது ஐஸ் கட்டிகள் வருதா, இல்ல புயல் இருக்கா, கடல்ல ஏதாவது மாற்றங்கள் இருக்கா அப்படின்றத அவர் பாப்பாரு. அதை பார்த்து அவர் என்ன பண்ணுவாரு, கமெண்ட் தான் கொடுப்பாரு டு த மெரினர்ஸ், அதாவது கீழே ஒர்க் பண்றாங்க இல்லையா, அவங்க கிட்ட இப்ப கேஸை ஸ்டாப் பண்ணுங்க, இந்த பக்கமா திருப்புங்க அப்படின்னு he will only comment. அந்த கமெண்ட்டை கேட்டு அந்த மெரினர்ஸ் எல்லாமே என்ன பண்ணுவாங்க, ஒர்க் பண்ணுவாங்க. கரெக்டா? சோ இந்த பாக்குற அந்த கேப்டன் தான் யாரு அப்படின்னு பாத்தீங்கன்னா, நம்மளோட மைண்ட். அந்த மெரினர்ஸ் யாருன்னா சப்கான்சியஸ். சப்கான்சியஸ் தான் நமக்காக வேலை செய்ய போகுது. பட் யூ ஹாவ் டு கிவ் யோர் கமெண்ட் ஆல்வேஸ் இன் அ பாசிட்டிவ் மேனர். ஸோ நம்மளால முடியும், நான் 680 வாங்குவேன், 700 வாங்குவேன் அப்படின்றத நம்ம மைண்டுக்குள்ள ரெஜிஸ்டர் பண்ண ஆரம்பிச்சோம்னா அதுக்கான ஒர்க்கை நம்ம செய்ய ஆரம்பிப்போம். அது நம்மளால முடியும். So don't ever think about negativity. Always think positively. That will always happen to you also. I hope you enjoyed the story. இந்த கதை உங்களுக்கு பிடிச்சிருந்ததுன்னா ஒரு லைக் அண்ட் கமெண்ட்டும் கொடுங்க. How was the story? Roger Beniter, yes, you are joined only for story, I think. Sanjay, enjoyed a story? எல்லா ஸ்டோரியில மூழ்கிட்டீங்களா? ரோஜர் பெனிட்டர், subconscious mind. Yes, you can. Yes, Jeevan, thank you. So இது எதுக்காகனா நம்ம சப்கான்ஷியஸ். எஸ், கிரேட், தேங்க் யூ விவேகான், தேங்க் யூ சுபான், எக்ஸலன்ட், என்ஜாய்ட். எஸ், ஸோ சுபானுக்குமே இது ரொம்ப பிடிக்கும். எஸ், நைஸ் ஸ்டோரி. யோகேஸ்வரி, தேங்க் யூ சோ மச். எஸ் சுபான், எஸ் தருணிகா, தேங்க் யூ. எஸ், இது இது எல்லாம் என்னால முடிஞ்ச ஒரு பாசிட்டிவ் நோட்டு தான் டு யூ பீப்புள். you are all young blood, so you can run more faster. இன்னும் கொஞ்சம் நீங்க டைம் எடுத்து plan yourself for the next 5 months. Okay, hardly 4 months. ஸோ அதுக்கு நீங்க பிளான் பண்ணி கரெக்டா ஒர்க் அவுட் பண்ணீங்கன்னா யூ கேன் அச்சீவ். நிறைய ஸ்டேட்மெண்ட் டைப் கொஸ்டின்ஸ் எல்லாம் எஸ் தருணிகா, ஆல் தி பெஸ்ட். டெஃபினெட்லி ஆல் வில் கெட். கீப் ப்ராக்டிசிங் மோர், அதுதான் அங்க ஸ்ட்ராட்டஜி. ஸோ படிச்சுட்டு நம்ம படிச்சுட்டோம் நீங்க நினைக்கவே கூடாது. பிகாஸ் யூ ஆர் நாட் கோயிங் டு ரைட் கான்செப்ட் பேஸ்டு கொஸ்டின் சில்ட்ரன், யூ ஆர் கோயிங் டு ரைட் எம்சிக்யூஸ். அப்ப நமக்கு டைலமா வராம இருக்கணும் அப்படின்னா practice, practice and non-stop practice till you achieve. அதுதான் இங்க வந்து இதுக்கான தாரக மந்திரம். And always refer NCERT first. அதுக்கப்புறமா நீங்க எந்த புக்கை வேணா படிக்கிறதுக்கு யூஸ் பண்ணிக்கோங்க. படிக்கிறத விட ப்ராக்டிஸ் பண்ண யூஸ் பண்ணிக்கோங்க. படிக்கிறதுக்கு ஆல்வேஸ் ப்ரிஃபர் NCERT ஃபார் பயாலஜி. ஓகேயா? ஸோ இன்னைக்கு நம்ம வந்து கிளைகோலைசிஸ் முடிச்சிருக்கோம். நீங்க இது கோ த்ரூ பண்ணுங்க. எனி டவுட் ப்ளீஸ் ஆஸ்க் மீ. And we'll continue the second part of the respiration in plant children in next week. Thank you all for participating. Yes, definitely Tarunika, support will be there. So please revise and practice more. Hi Rohit, very good evening. Almost the class got over, Rohit. I missed you the class. In the class. Okay, we'll see next week also. Respiration in plants is the 11th standard chapter, very important chapter. So I went very slowly. So you can join even for the next week in time so that you can understand. And your aim for aims will also get fulfilled. Yes, thank you, Subhan. Thank you, Jeevan. You're my motivator, Jeevan. Thank you very much. Thank you, Tarnika. Oh, it's okay, fine. Next week, let us see together. We'll meet. All the best for your exams, Rohit.